Our next presentation is Akadeum Life Sciences, and I'd like to welcome Brandon McNaughton, CEO and co-founder, to the stage.
Thank you. It's good to see everyone this morning. So it's been a year since we presented for our first time at Meeting on the Mesa, and a lot has happened this year for us, and that's what I'll be talking today about. But last time, if you remember, for those of you who were there, I started with a question, which is, could you put your hand in the air if you're aware or have heard of Akadeum? Anybody? Okay, so we have a good number in the audience. The goal is by next year to have everybody raise their hand. And we'll keep saying that until we have full awareness of Akadeum and the capability that we have. So if you ask, okay, well, what is the company and what are we about and what are we trying to do? Why do we develop the products that we've developed? And it's quite simple. What we care about more than anything is the quality of the cell that you use in your manufacturing process. That quality starts, in our opinion, on day zero, where you're doing separations or eventually activation. And if you've just looked at what's been happening in the field, and we have a lot of conversations with scientists around especially the US, and we've heard a theme. And that theme is we want to be able to isolate the cell that we want to engineer, say a T cell, and have it be untouched. Control the activation, control how you actually move forward with the cell. And what we've been able to do and what we focused on, and I'll be sharing more about this here in the slides, is we've essentially built the largest at-scale separation technology that can separate T cells and leave them untouched. So how we do that is we use tiny floating particles, we call them microbubbles. And what you'll see here is a video of those microbubbles. So you'll see a blue sphere in a second. So it's a solid particle, but it floats. You can put antibodies on them. They can attach and target any cell you want. So let's say a T cell. That T cell then floats to the top with the microbubble, while the other cells fall to the bottom. And you can either capture what you want with the microbubble, or you can remove what you don't want. So you can do depletion. You can negatively select the T cell, again, that's untouched. And what we've implemented and integrated this technology into is our automated and closed system. It's called the Alarion. That's what's on the left there. And it's quite large capability, about 100 billion cells an hour is the maximum with certain kits. And I'll show some data related to our scalability. And on the middle, what you'll see is something called the BioRise. So this is a consumable that each patient or donor sample that you have, you use one of these to essentially process that material and say, isolate your T cells. It's a two-chamber system, which I'll show you in a minute and how it works, but it's a very great format to facilitate either positive or negative selection for your cell therapy workflow. And what it enables, like I mentioned, is the scalability. We also care a lot about the gentleness and then also the yield. So those are three things we consistently see in our comparative studies and those done by our customers, the advantages that the technology has. To just dive into a little bit of data, and then I'll share a video of how this works. Let's talk about higher yield first. So like I mentioned at the beginning, a lot's happened last year for us. So the system you saw there on an image, that's been now commercial beta, and we're moving to doing evaluations and demos. So we've had the opportunity to work with a select group of customers to demonstrate what the technology can do and how it compares to other technologies. So here you see a yield comparison of T cells isolated with our system, and that's compared to magnetics. And what you see consistently across multiple runs and donors is a higher yield. So you get more cells that you want to actually engineer and start in your process. That data was done with collaboration with Poseida, so they were kind enough to do a webinar. So that is available on our website if you'd like to see that. The middle data here is really focused on gentleness. So the easiest measure of that is viability, but also the cell behavior can change too with harsh and not gentle processes. So here we see a difference in the viability of the cells isolated with microbubbles versus magnetics. So now you have something at your starting point, you have more cells and that are healthier. And that's really our goal all along, is to be able to provide that and then also to do that at a scale that's never been done before. And this is internally generated data, but what we consistently see is that pretty much across any scale of separation and even workflows, we maintain performance. So what that means and why that's significant is, say you're doing research scale work and you're using negative selection or getting untouched T cells, you see a candidacy that you want to move forward into your studies and preclinical. And as you scale up, there's no way right now to stay and continue to get untouched T cells and maintain negative selection. So when you get up to clinical scale manufacturing, you got to flip to positive selection where you're actually touching the T cell. And that affects it. And that's what we've seen time and time again. So we care a lot about being able to scale across any workflow and to maintain the performance and the gentleness. Across all of our products, we see up to 2 times higher yields. So that's for some allogeneic processes. So we have products that can remove dead cells at the end of the process, remove T cells. We see up to 40% higher viability, and like I mentioned, up to 100 billion cells per hour. So I'm going to just show again the BioRise, but now I'd like to show you a video of how we're able to do this and how we can essentially deploy the largest cell separation system to be built yet. So you'll see here in the video, the user will start by selecting a protocol. So they'll be selecting negative selection. So we're going to float out everything but the T cell. We're loading the BioRise. So it's a funnel-shaped consumable. We'll load one side with one chamber, which is clean media, and that's for the microbubbles to float into. You'll see it flip over. We'll add our sample. And then to the sample, we'll add bites-elated antibodies that are labeling all the cells but the T cells, because again, we're going to float those out. There's a 10 to 15 minute mix step. And then you'll see we'll come in with streptavidin-coated microbubbles to then complete the binding. And then now all that remains is to let flotation occur. So we do have a cell separation module. There's a slow rotational speed here to help create high reproducibility of separation. And if you look at the top there, you'll see a white layer. So again, that's where all the microbubbles and everything but the T cell went. T cells went to the bottom, and then with sterile tube welded in and draining into a blood bag. You can continue with your processes. So again, automated, closed system, and highly scalable and very gentle. So the kits that we have for this system, we've focused on really T cell first. We have other cell types that we have capability for, so that's NK cells, monocytes, PBMCs. But we were very intentional about saying, okay, let's start with T cells because they're commonly used and they're, for the most part, well understood in terms of these workflows. And so let's develop products that can handle those workflows. So we started with negative selection. So again, that's what you saw in the video where the microbubbles float out everything but the T cell. Those kits are available research use only in GMP. We have clinical ready work going on now. Our other 2 kits are clinical ready. That means we've done non-clinical animal testing to not only make sure we can make the material safely, but we can show and demonstrate that the material is safe. So the other 2 kits, we do positive selection, which includes activation and expansion. And like I mentioned, we also have products that can come on the back end and deplete unwanted cells. So when you look at the workflow, how we think about this is most of our products are either using autologous or allogeneic workflow. And so I know this is a generalization of both of those workflows. But when we looked at the products, we said, okay, where can we deploy this new modality of separation to help improve the process? So for autologous, we have the negative selection kit, and then we also have our positive selection and activation expansion kit. And that's something that we have users actively implementing right now in preclinical stage and research. And then as you go to allogeneic, same type of thing. We have the front end separation, but we can also come on the back end and deplete unwanted cells. So that could be dead cells at the end of the process. So that's been a very interesting thing, and their customers and our conversations have been, they need something that scales to high volumes and high cell numbers. And that's how they see our product and our technology because it's scalable at any volume. So you can come in no matter how big at the end of your workflow and remove those dead cells or even unengineered cells by removing T cells in a CAR TCR process. And as I mentioned, quality is a very important thing to us. And I know I talked a little bit about, what's happened since last year. We are in supporting one of our customers in its first clinical canine study. So that was exciting news to hear. We've also now started hosting customers for GMP audits. So that's another first for our company. I think that's an important point to demonstrate our quality, and that went well. And lastly, we also have our third-party manufacturing. So for customers who want to move into clinical trials and integrate our product, we have FDA-registered sites that we can work with for manufacturing the reagents that will come in contact with the cells during manufacturing. And all this really is with the goal of supporting our current customers entering clinical trials as soon as next year. So we have the product, closed systems, implemented quality quite aggressively, and been very successful in deploying that. So that's the instrument, that's the product that I mentioned, the reagents. But something else, I have a couple more slides here, but I wanted to talk for a minute about our technology outside of the Ilarion instrument. So one really neat thing about it, if you go back to the first video I showed, the microbubbles will just float in any size or shape container. So that opens up a whole new possibility, which is you can potentially reimagine your workflows because you can integrate it into existing instruments and use microbubble reagents to perform an important separation step. And so what that looks like, I mean, in its simplest form, you'll see a video here play. That's a time lapse of the microbubbles floating in a blood bag. And so you could actually do the separation in like a Leukopak bag. You could do it in a bioreactor. You can pretty much do it in any size or shape container. So the other part of our work, we typically have a lot of collaborations and partnerships on this, is to really demonstrate how that's done and the improvement that can make. So another webinar that's out there, it's not on our site, Lonza actually had a webinar and has an app note on microbubbles on the cocoon. So they've done some very nice integration work that you can see. And what's shown here is data that was generated using microbubbles and integrating them into the cocoon workflow. So that's something else we're very excited about. And we're also now exploring other systems that we could integrate into as well. Okay, just to wrap up, like I said, this for us is about really impacting the customer workflow, the scientist workflow, and ultimately clinical manufacturing. We care a lot about the cell and the state that it's in because we recognize if you don't have healthy cells starting, it's going to be really hard to end with healthy cells. So the better quality cell at the beginning, the better, in our opinion, and all of our products and technology were designed with that in mind. And also being able to be scalable unlike any other product out there. So what we're doing here today, you saw a little bit of our technology in our demo. We do have a table right out back. So if you want to see the BioRise or the microbubbles, we have actually microbubbles in a blood bag if you want to see how they work. Our table's back there. Andrew, I don't know if Andrew, if you raise his hand, Andrew's going to be at the table. His hand's up there in the back. And he has videos of the Ilarion too and information if you're interested in that. And then we also are here for discussions with partnerships. So Jill, are you still in the room? So Jill in the back there. So if you're interested in discussing integration, Jill's here for that. And as a company, we're also raising Series C. So I think we're here to pretty much talk to all attendees at the conference. I think we covered most everybody with that. I'll be available for a few minutes also at the table for any questions. Thanks for your time.